through so that way. Okay.
Cool. So yeah, thanks everybody for coming out to the Boston Protein Design and Modeling Club today. We have a special guest speaker that many of the locals here are familiar with, Sergey Ovchinnikov. So Sergey did his PhD with David Baker at the Institute for Protein Design in Seattle, and he's been a John Harvard Fellow here in, I guess, in Cambridge, Massachusetts for a few years now. And Sergey is really one of the, I would say, leaders in the protein structure prediction field. His PhD work really, he was one of the first people to use sequence coevolution for structure prediction. And I feel like this really laid the groundwork for a lot of the more recent advances that we've seen. And Sergey today is going to teach us how to use AlphaFold and RosettaFold using Google Colab notebooks. So Sergey, go ahead.
All right. Well, thanks you guys for joining. I apologize for my rather Rasputin-like look. I haven't been going outside much. So I posted, let me repost the slides just in case people are joining now. So if anybody wants to follow along or take notes or not. Also joining me today is Martin. He was working on me and trying to set up this Google Colab. So he'll join us a few slides later and describe that part of the project. Actually, Martin, do you want to say a word or two about yourself before we get started?
Sure. Hi, I'm Martin Steinegger. I'm an assistant professor at the Seoul National University. My PhD I did in the Max Planck and co-developed software there, like HHsplit, HHsearch, and like remote homology search methods, or like big clustering methods, search methods like MMSeqs. So really coming from the remote homology part, and yeah, for this presentation, I will also talk a bit about MMSeqs and HHsplits and how AlphaFold is actually building this multiple sequence alignments as an input for the model. Nice to meet you all.
Thanks, Martin. All right, so I think what I'll do is I'll start sharing my screen. So as far as today is concerned, what I was thinking is we will go through a few slides relatively quickly just to give sort of like the big picture view. Like we won't really go into the details of how the algorithms work. The idea of today's talk is more just giving you an introduction to what is AlphaFold, what is RosettaFold, and how can you use it, and just sort of more like a big picture overview. And so you guys may have already heard there's been a few papers that came out. One of them that I was involved in is referred to as RosettaFold, where we try to sort of learn from what AlphaFold did and try to build on those advances. And then the AlphaFold paper here, which made a huge impact during CASP last year. And one thing about both of these papers is that both, if you sort of stare at these diagrams, which I won't go into too much detail, is that they both have an input multiple sequence alignment. You have some multiple sequence alignment here, some sequence, and you search, you got a multiple sequence alignment, and you go through some kind of architecture and you output a structure. There's a lot of details in here, and so for today, I was thinking just giving it a big overview view. So the idea is, so typically the way structure prediction works is that you take a sequence and you search against a database of related sequences, and you construct a multiple sequence alignment. So the idea of like, if this is, let's say, a human sequence, you can gather a bunch of sequences from other organisms, and then you can stare at these sequences. You might be able to find some interesting patterns, like maybe conservation, maybe coevolution, maybe some relationship between the sequences. Typically that is passed to some algorithm to get contact predictions. And often these contact predictions are then passed to some folding algorithm combined with fragments. So this is sort of like the traditional way people used to do protein folding, is that you take multiple sequence alignment, you predict contacts based on sites that are covariant. You take the multiple sequence alignment, you construct a PSSM, you predict secondary structure, you search against the database of fragments, and then you recombine these fragments with some kind of folding algorithm, like like FragFold from David Jones or RosettaFold from David Baker. Alternatively, if you can take this PSSM and you search against a database of templates, you might be able to actually skip a lot of these folding steps. You might be able to just steal these templates and pretty much just copy those coordinates over and just replace the sequence. And so this is sort of like the overall pipeline. And there's been details and people try to put neural networks at every single step here. But each sort of step was sort of made advancements one step at a time. And one thing that changed with AlphaFold 1 is that, actually 1 second, is that instead of going through, if we step back a little bit, through a phase where we take the contacts and we try to fold the structure, one of the advancements was to sort of take the raw features from a Markov random field and pass them to a ResNet, some neural network, and the network would predict dihedral distances, and then you would sort of minimize structures to obtain that. So the reason why I have a little asterisk here is because there actually have been people that done these kind of steps before, but often like a specific arrow, but not necessarily the whole pipeline end to end to some extent. So these ideas have been floating around in the community, but it's just that each group sort of been focusing on like small parts of it. It's and then in some ways, one could argue that AlphaFold finally put all those ideas together to make this work. And finally, with AlphaFold 2, a lot of these things that were done, like I was describing here, like the fragment picking, sampling structures, and so on, was replaced almost completely by an end-to-end protocol. And where you have a multiple sequence alignment, you have templates, you feed them all into some kind of MSA module, you get some initial coordinates, and then you sort of refine these coordinates using a structure module. And once again, I have a little asterisk here because there were people that were experimenting with, first off, using templates as input to neural networks, but also trying to train these models sort of end to end. So like these ideas sort of been floating around. And also like even ideas of using sort of attention on multiple sequence alignments and so on. But one thing that remains across all these protocols that I just described to you is that all of them still contain a multiple sequence alignment. So these methods, even AlphaFold 2 and RosettaFold 2, that follows this very similar overall scheme, they still rely on the multiple sequence alignment and the templates. And this is sort of like our understanding of AlphaFold 2 was until recently when the paper came out and realized that there's another interesting step here where instead of sort of having some initial coordinates, you would actually initialize somewhere at 0, 0, 0. And then the structural module effectively just build up the structure from the center. And then you could actually feed the coordinates back into the network and sort of recycle and do these things over and over and over and over. And you get a better and better structure. So it's almost like a sampling procedure or a trajectory to some extent. And I'll touch upon that towards the end of my slides today. But the big picture, you're still depending on MSA and templates. And so the part that's kind of what we're trying to get at least in this presentation today, is that the predictions or the structures will only be as good as the input information. It turns out the template information doesn't play as big of a role. It's mostly coming from the MSA. And I think with that, I will let Martin take over.
Okay. Do you keep the presentation open and just move forward? I cannot
sure, that works, yeah.
Okay, perfect. So let me walk you through the AlphaFold input sequence databases. So AlphaFold is actually searching multiple databases, and it uses also multiple tools. So as Sergey mentioned, the MSAs are the most important, what you also see reflected here on that slide. It actually searches through 3 different databases. It searches through the UniRef 90. So it's like our public database. It's a clustered version of the UniProt and uses Jackhammer for that. But in order to find some more sequences that are really for difficult targets, sometimes in CASP, you cannot find much sequences for single targets. They search for metagenomic databases. One of them is Magnify, which is hosted by the EBI. It contains mostly human gut metagenomes assembled from the human gut, means mostly prokaryotic, also some viral sequences. And so they extracted the proteins from them. So it's much bigger than the UniRef already, I think with hundreds of millions of sequences available. And then there's the BFD. BFD was actually published with the AlphaFold 2 paper. That was the work I contributed to the AlphaFold 2 system. The idea is that BFD is from a different environment. It's mainly from soil, also metagenomic sequences. Soil is really interesting because soil has a huge amount of microdiversity. The biggest diversity comes from the soil. So we have developed an assembler that can assemble these really diverse environments, like get proteins out. So in BFD then collects all of these proteins, clusters them, and puts them into hidden Markov models, so they are searchable with HH splits. And also you can search them iteratively with HH splits, meaning you can go through, you find a few hits, you build some model from that, and then you search again, and you get more sequences, and so you can make bigger and bigger MSAs. And on the right side, you can see the PDB-70, which is used for the template features, but as Sergey already mentioned, the templates are not that important anymore, actually, for the performance of the AlphaFold system. The MSA is way more critical to use it to get a good prediction. Could you switch the slide, please? Yeah. And the AlphaFold currently one big issue with AlphaFold is if you want to install it on your local cluster and you want to run it, and these databases are really huge. So there's like 2.5 terabytes or so in space you need. And to search through them on multiple cores, you need hours for a single protein. So it can take hours. So it's really heavy to search through them and build these MSAs. Okay. So in ColabFold, we wanted to see if we can replace this heavy stack of jackhammer and HH blitz with a method called mmseeks2. So mmseeks2 is a fast search method that can also cluster sequence databases, but here we'll talk mainly about the search aspects of it, which has a very fast pre-filter, and the pre-filter has the task to search through big databases really fast and like reduce the amount of candidate sequences as fast as possible. So we use some kind of like k-mer based pre-filter. And could you click, please? And the idea for that k-mer based pre-filter is that we use quite long short words, like short k-mers, and not just exact matching, but also similar matching k-mers. So we try to go through the query sequence. We find our k-mer matches, and for our k-mers we build lists of similar k-mers, and then we search them in the target database, and we find proteins where two of these long k-mers, where in one diagonal, two of these k-mers occur on one diagonal, so like double diagonal hits actually. And once we find a sequence that has a double diagonal k-mer hit, we do in full alignment, like doing expensive Swiss Waterman alignment for that sequence. Could you click one more time, please? And sorry. Yes, to mention, there are many fast search methods out there, but mmseeks2 is the only fast search method that can actually use profile information and can also do this iterative profile searches, which is really critical to build these multiple sequence alignments. Yes, so here in that plot, there's a comparison between mmseeks2 and other search methods. Maybe you will be familiar with probably BLAST. Swipe is a Swiss Waterman implementation. These are the fast ones. You can see them here in the black circle and green square. So in this plot here on the x-axis, you can see sensitivity. On the y-axis, you can see the speed up. And on the left side, there are the sequence comparison methods. And on the right side, there are the profile searching methods. And you can see already that the profile searching methods are much more sensitive. Here, these small numbers indicate actually the iterations that were searched against that database. And you can see that mmseeks can be, I think, around 300 to 400 times faster than PsyBlast by being even more sensitive than PsyBlast. And there's no other method in that segment that can really keep up with it. And one more important thing, that's where Milot Medita comes in. He's also part of that project. he built a server version for MM6, and that server version had the task to reduce the runtime for single queries. So with that server, we can actually search single queries in a few seconds. So we can go through these really big databases, and it takes only a few seconds to search through them, which is really crucial here for handling all the requests that we get now with the ColabFold. And yeah, so we wanted to see if the MM6.2 version can actually compete with the other versions of the AlphaFold system out there. There are currently two versions. One is the DeepMind Colab. So they also built a Colab system. So we published it, actually released it, I think like a few days before them. So their Colab system is using Jackhammer and searches through a reduced database. Then we also wanted to compare it with our AlphaFold 2, the full system. And what we did is we took from the last cast, we took all of the FM targets, free modeling targets, so the ones that are supposedly very difficult. And we ran it through their Colab and our Colab and the full AlphaFold system. And then we checked how well do these templates, how well do we can predict the structures, and we compare using TM align. So it's a score from 0 to 1, where 1 is the best. And so then we just plot these two dots for example, on left side from the DeepMind Colab and the ColabFold for MM6.2. You can already see that we perform fairly similar to the to their Colab system. For some targets a bit better, but overall pretty much the same performance. For the AlphaFold 2 full system, we perform also very similar, aside from these three blue dots here on the bottom. These colors here indicate actually how much sequences the AlphaFold system could find for each of these targets. Meaning for blue you have less than 30 sequences in the MSA. So meaning if you don't have enough sequence information in your MSA, then you cannot predict these targets very well. And actually some of these targets that have quite a big difference here, the one here on the right side, has only, I think, 2 or 3 sequences. So the AlphaFold system only found 2 or 3 sequences, and that was enough to get an TM score of greater 80, so 0.8, which is pretty impressive, but it means like sometimes only a few seconds can make a big difference for a target. But overall, I just want to show that the system that we have built can actually keep up with the systems that are out there. Maybe can you click one more time, Sergey, please?
Mark, could you briefly mention just like the speed of this versus this versus
yes,
this?
Yes, that's what I wanted to.
Oh, is that the next slide?
I have it in a remark. Yeah.
Okay.
So, so the DeepMind Colab takes for one target to search with the Colab can take also an hour through using Colab, while the AlphaFold system, as I mentioned, uses multiple cores and also takes an hour, while the MSA generation for here that I show 20 sequences takes with MM6 takes only 4 minutes on a single core. So it's much faster. This enables our Colab to make predictions extremely fast in comparisons to these other systems. So submit something, sometimes you can get a structure back within 10 to 15 minutes. So the bigger part here for us is actually running the prediction on the GPU, while before the problem was getting the MSA, like taking an hour to get the MSA, and then the prediction took like 15 to 20 minutes or so, and now it flips around. Now the prediction on the GPU takes much longer than the input MSA generation. Yes, and as I mentioned before, these metagenomic databases are kind of crucial for sequences that are hard to predict, especially like viral sequences or some really rare archaeans or so. So in the MM6 server, we currently support BFD. We used a reduced version of the BFD. We added the magnifier as well. Also, these magnifiers the same what the AlphaFold system has. But we are not searching through the whole set, but we search through a reduced set. So we are just clustering all of these sequences down and just take the representative sequences from them. We added some more sequences into the mix. One is from MetaYuk, These are eukaryotic proteins. Sometimes it's really difficult for some of our big eukaryotic organisms to find enough protein. So we thought it might be useful to add some more. We also added this SMAC database. It's also eukaryotic proteins. They're mostly from oceans. Could you click one more time, please? And we are planning to add more and more sequences to the server because the MSAs are so crucial, right? So the better your MSA, the better your prediction, and sometimes you cannot find things. So we wanted to make the best possible predictions. So we search for more metagenomic databases out there. So coming soon, we will add TopPass. It's also eukaryotic proteins, also from marine environments. We added viral metagenomic databases, and a big phage protein database, and one unpublished database that consists of 36 billion proteins, which we have clustered down now to only a few 100 million proteins. And hopefully we will release that in the next few days. And we'll keep you up on Twitter and posts about it once that is online. We also want to evaluate if the cast performance improves on that, or if we break something. But once that is done, then we will switch to the new system, and hopefully the MSAs will be much better. Okay, that's all. Thanks. We'll back to Sergey.
Thanks, Martin. But I guess before we switch back to me, does anybody have any questions for Martin? You can speak up, and Martin will try to answer them.
Somebody asked what is essential for running the full AlphaFold system. So since AlphaFold consists of these two parts, the first is the MSA generation part, and the second one is running that model. Actually, the hardware is heterogeneous. So for the search, you would want to have a lot of cores and RAM available. Probably you can run it with a system that has 128 gigabytes of RAM and like a few cores, then the search will probably work fairly well. And if you store the databases on an NVMe disk, then it will be also much faster. So I think like using NVMEs would be quite useful. And then for the GPU, the RAM of the GPU is the most important for like big models. The more RAM you have, the faster it can compute. But for example, an RTX 3090 would be a very good thing to do predictions with. It's probably not that expensive to put it into your computer. Yeah.
I had a quick question. I was wondering if you can pre-compute MSAs in some, I know that you can't always know what the query sequence is going to be, but maybe you can know a related sequence to something that, or even cache, something that people frequently request?
Yeah, so in the server, we are caching queries that were already submitted before. And so if somebody has already something, then we just return the same MSA. But what you're talking about is a bit what actually profile HMMs like HHBlitz and so does. What they do is they take the sequence database, they cluster it into 30% identities, then you end up with protein families or something like this. Each cluster could be a family. Then you build an MSA for each of these families. And what you now align, you only align these families, and then you extract all of the sequences that are within one cluster when you hit that cluster, when you find it. So yeah, that works. Also, there's the idea of like you take the clusters and you search them against itself again, and you can make the clusters even bigger and have bigger MSAs and can extrapolate more sequences if you want to. I think this is pretty much going in this direction of like compressing the MSA space. Yeah, so with MM6, we do something similar. We take the UniRef, we take a clustered version of the UniRef, searching against the cluster centers, and also extrapolate the members of the clusters, which makes it faster because we don't have to search through like the full redundancy and finding the same sequence thousands of times or hundreds of thousands of times sometimes. Some more question, is there a general resource reference that describe MSAs and why they are so powerful, sort of intro to MSAs reference? I'm not aware of a good reference one where I say MSAs are very powerful in biology. I mean, they are shown to be really powerful in many domains, building trees, making the co-evolutionary studies and things like this. But I cannot point you to any specific paper for that. Do do you plan to add databases from the JGI? So the MetaClust 2 database I have mentioned before is from the is part of JGI proteins. So they're for MetaClust 1, we're going through the assemblies, extract assemblies from the JGI, metagenomic assemblies, extracted proteins, and built this MetaClust database. For MetaClust 2, we did the same, but like with all the updated new sets, which resulted in this like I think like around 36 billion protein sequences. Okay, and one question again is if you can explain MSAs on a high level, what do these databases provide us? So MSA on a high level. So you can imagine you're starting with your query protein, right? And now you're searching through the database. And what you do is you find homologous sequences, so things that are in evolutionary time somehow related to that query sequence. And what that MSA actually represents is the relationship from each residue of the sequences in the database to your query sequence. So you try to find which of these residues are evolutionarily related to each other. So the MSAs at the end, each of the lines is one sequence, right? So you have many sequences, and each column is then one residue. And a good alignment is one where you find all of these homologous sequences that are somehow related to the sequence you're interested in. And the columns are also like each residue is correctly aligned, meaning that is really the origin of these residues. And then if you have that, that gives you like an evolutionary picture of how the protein has changed over time. And Sergey has mentioned like these co-evolving columns, which is used for contact prediction. In this MSAs, if you look at the columns, you can find like residues that are evolving at the same time, and they are often in physical contact within the protein sequence. And that's really what the AlphaFold system probably also reasons about find these columns that are co-evolving and having these contacts.
Yeah, those are really good questions. I put in a few slides that I can tell you a little bit about MSAs for those of you who prefer a visual here. But before I switch to that, any final questions for Martin? I guess there's one question related to like, are you planning to include sequences from JGI?
Yes, I have mentioned that. Yes, we are. The MetaClust 2 is based on the JGI sequences. And there's one more question. I think the question is the depth of the MSA is really a good metric. Yeah, it's a good question. It's the depth of an MSA is by definition not good, right? You can imagine you take an MSA and you just take the same sequence 100 times in the MSA or so. That's not giving you any information, right? So it's a metric of like how diverse is that MSA? Meaning if you reduce redundancy from that MSA, how much sequences survive this redundancy filter? And then you count. And also the coverage is important, right? And sometimes you have a multi-domain protein. If you have only hits in one part of one of the domains, then AlphaFold can model that domain very well. But then maybe on the other side, you don't have any information. So the raw sequence count is probably not always the best. You want to see if you have a like good coverage over the whole sequence that is diverse enough, and then the system can predict things very well. Okay.
Do you have, if I may, do you have any reference to what would be a better metric then? If just like line counts are bad, is there any metrics that developed to show that's a good MSA? I
think Sergey, in your metagenomic paper, you suggested this NF measure. I don't have it exactly in my head. It was something with coverage and redundancy. Maybe can you explain that?
Yes. So I guess on the big picture here. So what happens is every method kind of depends on different number of sequences. I mean, what we found was that we needed like at least 1,000 sequences in older methods. But one thing that DeepMind has found with their recent method, AlphaFold 2, is that getting 30 sequences often is enough. And after you go beyond 100 sequences, you don't actually add too much more information, which was actually quite exciting because normally we thought we needed at least 1,000. But one thing I should mention is these 100 or 30 sequences, they can't all be identical to each other. So they have to be rather really diverse. And so I think one way, since we do have some people here that probably haven't thought too much about MSAs before, I think when I was putting these slides together, I kind of assumed you've seen all my previous talks, but I guess we have a lot of new people here. So let me see if I can show a few slides that I think might help get people up to speed what's going on. So I guess one of the question is, what exactly is an MSA, and why did we invite Martin to talk about MSAs? Ignore this title for now, but the general idea is imagine that you have a sequence. This is, let's say, a human sequence, and you might stare at the sequence, and if you know some biochemistry, you might look at the different characters here and say, well, this is a hydrophilic amino acid, this is a hydrophobic amino acid, so maybe you can guess somehow how this protein will fold up. So this is a string of amino acids. But it turns out what you can do is, instead of just looking at one sequence, we know that all organisms are somewhat related to each other. And so what you can do is you can take this single sequence and search against a database of other sequences and construct a multiple sequence alignment. And the reason why a multiple sequence alignment is quite powerful is because before you just had a single sequence and you don't really know which are the most important characters. But once you construct a multiple sequence alignment, you suddenly see there's one position that is the same across all the different organisms. And then there's two positions that appear to be covariant. Like every time there's a change here, it appears that there's also a change here. But the other site here may tell you a little bit something about the relationship between the sequences. And so things like this is what now gives you a lot of interesting, powerful information, because now you can sort of use this information to tell you that maybe the sequence actually has some kind of constraint, and then you've been able to fold up the structure using that constraint. And so this is sort of traditionally how people have used multiple sequence alignments. And we suspect that neural networks that take this multiple sequence alignment as input are looking for this exact same pattern. They're just looking at the sequence and they're like, okay, these are really important positions. These are positions that are covariant. So I guess on the computational side, you're potentially reducing the search space because you're suddenly adding these constraints that were otherwise not visible. So I think that's why these becomes quite powerful. The other thing about, as far as these multiple sequence alignments are concerned, so the other thing that we didn't really talk about is phylogeny, and we think that AlphaFold also sort of took this into account indirectly. And why this becomes kind of important is because you might imagine there might be a group of sequences that, let's say, are coming from a cold environment, and there might be another group of sequences, let's say, that are coming from a hot environment, and you might actually have different patterns of covariation going on. So for example, maybe this forms slightly different structure than this forms a different structure. And so for example, let's say if this is the protein that you're trying to actually model, you might be able to use some of these sites to tell you, okay, these are the sites that are most important. So there's a lot of things, you can sort of capture the evolutionary pressure from these multiple sequence alignments. Looks like somebody. So one of the questions is, what do you mean by indirectly account for? We didn't get too much into the details of the architecture they used, but one thing is you can think of this MSA. So if we just step back for just pretend that this MSA is just a picture. Just imagine that this is just some kind of image here. This is rows and this is columns. And what happens is this image gets passed to an attention mechanism, and the attention mechanism looks at the relationship between positions, but it also does another attention that is actually the relationship between the sequences. And so by indirectly, what I mean is it doesn't actually construct, it doesn't explicitly make this kind of tree, but it does sort of understand sort of, you could maybe potentially approximate this tree as a bunch of maybe connections. Say there's a connection here, there's maybe a connection here, maybe there's a connection here, and so on. But the idea is you could sort of learn the relationship between the sequences maybe in some kind of embedded space as opposed to directly a phylogenetic tree. So Duka Extra is asking, does the fact that AlphaFold needs fewer sequences suggest that it does not rely on pairwise contacts? That's a really good question. So the thing is, it still relies on sequences. It's just the idea, the reason why we think it relies on less sequences. So if we step back a little bit, let's go back to their overall architecture. What's happening here is there's this evoformer block that takes in that MSA and effectively processes it 48 times. And at least the way I like to think about what this evoformer is doing is sort of an unrolled version of these coevolution algorithms. So normally the way we would do coevolution analysis is we take these sequences and look for covariance patterns and then try to infer which residues are in contact with each other. But the advantage of sort of with this kind of block here, and the same thing with even the Rosetta fold, is that you're sort of progressively trying to both extract the context from the MSA, but also you're introducing sort of a structural prior. So the idea would be, so one way to think about it is like you're trying to figure out the coevolution, and you're trying to also include the structural priors that will tell you potentially, and so that might actually give you require less number of sequences to actually pull out who's in contact with who. One thing that's been actually quite exciting is that after both Rosetta Fold and AlphaFold got released into the world, there's been a lot of people that sort of took these algorithms and took notebooks that we made and other people made and sort of started effectively doing research live and tweeting them as soon as they found something cool. So this is a bit unprecedented because I think often people, when they find something cool, they rush to write a paper and hide it from everybody. And then maybe a year later, we find out what they did. And then there's like 10 other people that did the same thing. Now everybody's just on Twitter, just going crazy, shouting out all the cool stuff they found, which I think is actually kind of exciting. Just to give you a few examples, there's been people that sort of built models and were able to solve structures that they were previously not able to solve. So they had some X-ray density and they were not able to do molecular replacement, and people were like, I just ran this and I was able to get a solution. Somebody's just growing. I say this is much easier than growing crystals. Also, people have been using these models to potentially now fit into cryo-EM density. So this is more maybe what I would consider to be like more of an intended use. Like you can use these models to now potentially start to solve things that were previously unsolved. But also there's been some really interesting sort of unintended uses. So these models, these AlphaFold, Rosetta Fold, were not really designed to predict protein-protein interactions. But one thing that people have found was that, hey, if you just pretend the two proteins are just a single protein, you just add a gigantic glycine linker, it actually predicts that protein interaction almost correctly. I think there was other people discovered like actually instead of a glycine linking, you put like an unknown linker, and that also works. And also you could do like protein peptide interactions. And Ming-Yang went on to say even further, well, actually you can even hack the AlphaFold code and just tell it that these residues are really, really far apart, and it actually treats them as sort of separate molecules. And so one of the cool excitement that's been coming out was that it looks like we might be able to just take the network without even retraining it and just start predicting protein-protein interactions. Just to give you a few more examples, I think there's somebody's like, oh, it can do cross-species interactions. It can understand dimer swaps. It can model intertwined dimers. It can model peptide protein interactions. And I think there's even, people are now beginning to even publish some things on the preprints, which kind of brings up an interesting question, like who should get like the credit, like who first tweeted it, who first preprinted it, or who got it first published, which is, I guess, something we can debate in the future. So there's been a lot of interesting sharing, and I'm guessing a lot of people are, but besides protein-protein interaction, there's also been some really interesting stuff where instead of predicting a single model, you can actually take all the different trained models and output the structure, and it looks to be capturing some kind of intermediate structures between known conformations. I think Kristen was saying like, oh, actually these models could have been able to sort of validate like which of the crystal structure is the correct crystal structure. Sorry, not crystal, but like NMR structure. And also potentially use these models to do stability prediction, and also even some of these metrics coming out of these models in terms of like how confident the model is about different parts have been able to be used to potentially predict disordered regions in the proteins. Shane asked, will antibody complex ever be picked up? That's a good question. So after today, hopefully you'll be able to submit the antibody sequence and you'll find out. Okay. And just ... Now we're moving on to the next section. Is there's been a lot of excitement, and one thing that I've been doing is sort of like looking at Twitter, and every time somebody comes up with something cool, I've been trying to sort of incorporate the notebook so other people can try. And so one of the things that was found actually kind of exciting was that it appears to even be able to predict homoligomeric structures. So if you just take the sequence and literally just make a bunch of copies of that sequence and just pass it to AlphaFold, it could actually predict correctly the agglomeration states. Here's like EMRE, which is a transmembrane protein, was able to also predict that correctly. But one thing that was actually kind of interesting, and this is kind of the puzzling part right now, is that if you take the let's say you have a multiple sequence alignment, a bunch of sequences that are related and a bunch of sequences related for the second protein. I mean, in this case, it's the same protein, and you just concatenate it this way, it doesn't work. But if you provide it like this, like you say, I'm going to provide the first sequence of all my data concatenated together. So this is the first copy of the protein. This is the second copy of the protein. And then you provide a copy of the MSA here and a copy of the MSA here, and that actually does work. And so it's kind of interesting that this doesn't work, but this does work. And then Ming Kyung suggested actually you can go in the source code and modify to add links here, like just add a large residue, count here. So the idea is one of the input features to AlphaFold and RosettaFold is just the residue index. So this is like 1, 2, 3, 4, 5, 6, and so on. And so you could potentially just say, okay, this is my second protein, so I'm just going to now number this as 200, 201, 202, and 203, and so on. And that's enough to trick the system into modeling these things as separate proteins. And the other part that's actually kind of interesting here is that if you just model a single monomer, so this is an example where we modeled a single monomer. This is the pink protein, and this helix, and the correct answer, sorry, is the blue and the green. And so you can see this helix actually go in the wrong direction, but if you actually model it as a homodimer, it then actually correctly places these two helices. So it's not necessarily that this is just recovering some kind of higher order things that the model already learned and is just waiting to be rediscovered. It appears to actually putting in the information that these two proteins are together allows it to actually fix some of the conformations of the structures. And the other thing that's actually kind of interesting is that if you give more copies than there actually are, so for example, this is a protein that's a homodimer, but if I give it like 4 copies, it actually puts these together, but then it doesn't actually try to predict that these two interact. So this is where I'm going to quite quickly introduce an interesting metric. So one thing that both RosettaFold and AlphaFold provide is some kind of confidence metric. This is predicted LDDT. I won't go into details what this actually means, but the idea is like, you could think of it as just a confidence metric. So 100 means that you're 100% confident about this. 0 means that you're not so confident. So anything below 50, you would say like it's very, very low confidence. So for every single residue, you have some kind of confidence metric. And on top of that, for every residue pair, there's also this predicted aligned error that's provided by AlphaFold. And so here what's happening is you could see this is the first protein. This is the green guy is going from residue 0 to 100. The blue guy is residue 100 to 200. And it's high confidence predicting that these two are interacting with each other. Oh, and by the way, this metric, you can almost think of it as sort of like the predicted variance or an angstroms. So, and here's a color bar here. So 0 means that it's really, really confident about every pair of interactions within this little block here. It's also very confident between like the interaction of this protein and this protein, but it's not confident at all about the interactions between, let's say, copy 1, 2, and copy 3 and 4. So this will become really, really important when, especially if you're assessing oligomeric predictions, or if you're assessing even just domain-domain interactions in a very large proteins. So both of these metrics are provided both by us and AlphaFold, and RosettaFold also spits out this metric as well. But one caveat here is like sometimes it actually doesn't work, and so you have to be a little bit careful. So here's an example of a protein that's actually a 6-mer, a homo 6-mer. And so if you ask the algorithm to predict the monomer, it predicts it as such. You tell the algorithm to predict a dimer, it predicts it as such, and it tells you it's pretty confident about the dimer-dimer interaction. If you tell it to predict a homo 4-mer, it's also able to predict it. And then if you tell it to predict a homo 6-mer, it's able to nail it exactly. So this is ground truth here overlaid on the correct structure. But the thing that was kind of interesting, and this is a little bit depressing, is if you tell it to predict a homo 8-mer, it actually still predicts it a homo 8-mer, and it still thinks that it actually interacts. And so just a word of caution is that if you give it too many examples, sometimes it's able to really, like if you, in this case, I gave it too many examples and it says, okay, these two are separate from these two, these guys interact, and these two do not interact with each other. But sometimes, especially I think in cyclic symmetries, like when you have actually sort of like a repeating cyclic symmetry, like in this case, like if you look at it actually like this interface, like this sheet comes out here and pairs up with this sheet here, this beta strand here. And here, like this interface is actually not much different, like as far as this is concerned. And so it looks like it's not really able to tell like the difference between this guy and this guy. I guess one could argue maybe in solutions, sometimes this one forms and this one forms. So who knows, maybe, because usually the crystal structure is like the most dominant conformation. And that's what we see in forming in crystals. But maybe there's a fraction of proteins that actually form like this. So it might actually be real. But yeah, just a quick warning that it doesn't always work. It looks like it does really good job, but if you have a cyclic symmetry, it might actually put more copies than there should be. And I mean, the idea of sort of using these algorithms like Rosetta for AlphaFold, predicting structures, has actually been described in the Rosetta Fold paper, and this is actually one of the figures from that paper. So it's not necessarily completely brand new, the fact that it can do this. I mean, this is what we literally did in the paper. But the fact that AlphaFold can also do it, which is something that the DeepMind did not describe, was something actually kind of exciting. But one thing that makes it kind of really interesting and unique is that in Rosetta Fold, in order to get these complexes to be predicted correctly, you need to construct a paired alignment. So you need to have a bunch of sequences, like say from protein A and a bunch of sequences from protein B, and you have to provide this multiple sequence alignment to Rosetta Fold, and Rosetta Fold is able to predict the structure. The part that's kind of surprising and still kind of a puzzle is in AlphaFold, even if the sequences are unpaired, like let's say you have a bunch of sequences for A and a bunch of sequences for B, and you don't know how they're paired, like which genome they came from, you could just literally provide them like this, just staggered, and it still works. It doesn't always work. So Ming-Yang found a few examples where actually adding a few paired sequences made things a little bit better. But surprisingly, it actually seems to work more often than we saw Rosetta Fold, for example, for the same data sets. It seems to understand something about maybe crystal packing. I think there was one post that I forgot to share where somebody noticed that it appears to be capturing sort of crystal packing contact as well. So not just the biological units. Like if you give it multiple copies, it will actually form the crystallographic assembly. And the other thing that's kind of interesting is I think there's been one group that initially proposed using these gigantic glycine linkers. And this is for one of the SARS-CoV-2 proteins. It's a homodimer. And what we see is that actually not trying, so instead of trying to hack the algorithm by putting a giant glycine linker, not including it actually gives you better answers. So in our, for example, our web server, we do not use a glycine linker. We actually just use that trick that was I previously described from Ming-Yang of just modifying the residue index of the protein. So telling it, okay, these are two separate proteins and they're separated. And it turns out that doing that actually works a bit better. In this particular example, it was able, in this particular example, when you included the linker, it actually, it looks like it messed up with the whole folding or assembly of the protein. Okay, so now adding one more interesting observation, and I guess if you guys are following me on Twitter, you might have seen me talk about this a little bit. This idea that what AlphaFold does is that instead of just going through the network once and predicting the structure, it takes the structure and converts it into a distance matrix and converts into some C beta bins, and then converts those into pairwise features and feeds it back into the model. In some similar way how you sort of feed in templates to the model, you could sort of feed in a prior prediction back into the model, and then let the network say, let's try again. And then you feed it back in and say, let's try again. And each time you're sort of iterating and you're potentially making the prediction better and better. And also the pairwise features that just come out of the model also being fed back in. And by default, this recycling is only done 3 times. And it seems to be sufficient for most cases. But one thing people have noticed is if you actually try to predict oligomers, and this is a credit to Ryan, who shared this slide with me, that sometimes doing more than 3 recycles actually gets you the correct conformation. So in this case, I believe he needed about 10 recycles in order to get this oligomer to form. And so the idea is like so the first picture, all of them were, that's recycle 0, they're all just overlap with each other. And then the second recycle moves it in further and further and further until they finally all come together. And so the idea is like, as you're feeding in the models back into the MSA module, they get processed again and feeding it in, each step actually appears to be refining the model further and further and further until if you run it long enough, eventually it converges, and in this case, you get the right structure. Besides just homo-oligomeric assemblies or complex assemblies, turns out even actually helps in the context of monomeric structure prediction. So this is an example where this is a protein that was mispredicted at recycle number 3, and this is an example from Anastasia. And so she, she told me, hey, AlphaFold doesn't work. I feed in my sequence, it didn't give me the sequence back. And so I tried to increase the number of recycle units, and actually after recycle number 4, you got a much closer structure. And then after recycle 6, the structure was almost dead on. So it turns out that adding more recycle does actually help. And here's actually a kind of a cool example where it's literally, this is a case where I actually had to run for almost 12 recycle units, 12 recycles before it's able to correctly fill up the structure. So here, what I'm showing here is the structure at each recycle. Here I'm showing the predicted LDDT. This is the confidence. And as it's running, it's not only getting closer to the right answer, it's actually becoming more and more confident about the answer. And here I'm showing that same structure, but now colored by the predicted LDDT. And so this whole recycling appears to not just make the structure better, but also the cool thing is that the model tells you when it is confident. And that's actually one thing that I forgot to stress about both RosettaFold and AlphaFold is that when it doesn't know, like it's not sure about the answer, it's actually really good at telling you that it doesn't know the answer. So it doesn't just give you a prediction. Like if it doesn't know, it will give you a structure, but it will give you a low confidence for every single position. And so you know, okay, there's not enough information, or maybe I didn't run enough cycles or so on. And so that's one actually kind of interesting thing about these algorithms. The other thing that's kind of interesting is that if you change the seed, you might actually get a slightly different fold. This is, for example, with seed 0, 0, you got these 2 sheets here, and then with seed 1, these sheets flipped over. So, so it looks like even, so one thing that I forgot to mention is one of the inputs, so the MSA, it's sort of being clustered and then passed to the MSA module. But there is a random seed that's involved here in the clustering aspect. And so it turns out if you change the seed, it will give the input feature slightly perturbed. And these input features perturbed may give you actually a different structure. And it's really confident about both structures in this case. So if you want some more structure variability, looks like the seed might actually be able to help. Okay, so I think with this presentation, I'd like to end here. Some people to thank from RosettaFold, they helped me sort of made sure that the algorithm was running correctly and so on. The AlphaFold people, I consulted with Tim and John to make sure we're running the algorithm correctly. Inzu submitted a few bug fixes, and Lim actually did some of the analysis, some early analyses. And also David Coase, because without him, all of our notebooks would be really boring because it would just be code. But with David Coase, we can use his library to actually display the 3D structure directly in our sessions. So I think the visualization is actually just as important as I think any of this stuff. And finally, our funding sources that all of us involved got money from. So Samana asked, would running cycles in Rosetta 2, would that actually help? Yeah, that's a good question. So we saw in some of our analysis that recycling also does help improve the models, but we didn't train in the context of the recycling, I don't believe. And so we think that maybe actually training in the context of recycling may actually help. This is one of those details that when AlphaFold or DeepMind first presented the architecture, they didn't really, they left a few, I think, key details out, such as the recycling step, and also for example, training on predicted structures and so on. And so we think those are sort of critical parts that we have not yet incorporated in RosettaFold, which I think may actually help us catch up, or maybe even do better. We'll see. So another question is, how many cycles are needed before you stop? So the interesting thing is, it turns out that as you just keep running, eventually it just converges and stops changing. And so one thing that I've implemented in the notebooks, which I'll show you in a little bit, is that you can just set to some very large number, and that'll automatically stop as soon as the cyclings converge. I think I addressed most questions.
Okay,
if somebody has another,
maybe I'll ask a quick one. Disulfides. Is there a way to form disulfides, or is everything under reducing conditions right now?
So it does form disulfides if they're present. Like, it brings the amino acids close enough that the disulfides will form. And it does, I think AlphaFold showed in their papers that even though it's not modeling, for example, zinc or heme or any kind of ligand, it actually, if you look at the side chains, they're almost perfectly set up to handle those ligands. So it appears to implicitly sort of understand that there is something there. So Ava is asking, what about de novo designed proteins? Do you think generally the predictive power is strong enough? So similar to what we found with TR Rosetta a while back, is that when you give it a single sequence, it actually surprisingly works really well. And we're finding the same thing to be true with Rosetta Fold and AlphaFold. But one thing I should say is that I guess one thing I forgot to mention is this right here is an example from Anastasia. And this is actually a de novo designed protein. And there's literally only one sequence that gets fed into the model. And what we see is that whenever there's a very limited number of information, this is where the recycling actually plays a big role. So the idea is, if you have a lot of MSA here, then often even just doing a single recycle is enough to get the right structure. But if you have very noisy data or very, very sparse information, or even literally just a single sequence as the input, then you do sometimes need to vary the seed number. You need to vary the number of recycles in order to predict the structures. But also these are somewhat unique examples because these are transmembrane beta barrels. Okay, I guess we'll come back to that. But Simon asking, is there any risk of inflating the confidence score? Oh, lower limit. Okay, sorry. I think sorry about that. Okay. Yeah, that's a really good question. So I mean, these observations that we've seen and people have been posting on Twitter, one thing I would caution is that these are just a few examples that we've all kind of looked at, and we saw some really cool results for individual cases. I mean, the future will be all about benchmarking. Yeah, so there's definitely a danger that maybe if you do too many things, you might artificially make the LDDT really high. And maybe that could be some form of, like, as people say, like R hacking or something, where you do like crystallography and you sort of keep changing things until your R-free looks a little too good. So yeah, there's definitely some danger, and I think we should be careful with that. But so far, what we've seen is that whenever the predicted LDT is higher, even with more recycles, that it actually does correspond to more accurate structure. And in this case particular, the one that I was just showing here, this is a case where after doing a lot of recycles, the confidence got really high. But actually, if you try it on other proteins, it doesn't actually go up high. It stays pretty low even if you do more recycles. So it looks like it's only goes high when it becomes actually more confident. So the question is there any issues with Colab running? So unfortunately, every recycle is going to increase the runtime, but the runtime is linearly increased. So if you do 2 recycles, it's going to run 2 times. It's going to take 2 times more time. I think Firas mentioned that AlphaFold 2 is now integrated and folded, which is kind of cool. You guys should definitely check that out. Okay, so I think since I took quite a lot of time on this presentation, and I guess I promised some tutorials, so let's go ahead and show you guys a few tutorials really quick. If people need to run, don't worry, you guys can escape. We will post this video later, so you can always watch. So if you need to go and either go to sleep or wake up, no worries. Okay, let's see here. Okay, so the big picture here. So we do have a GitHub page, SoCrypton at Colab. here. And this link I will post. Let me go ahead and post that in the chat box really quick so people can see it. And so in this note, if you guys have been following me on Twitter, you probably noticed that I've shared a lot of different notebooks, and I think it probably got a little confusing because maybe I shared a little too many different ones. I try to sort of organize what the pros and cons of each notebook is here. So if you're like, hey, I don't know which notebook I should be using, here is sort of a sort of checklist, I guess you can say, what each of the notebooks does and what they offer and they don't offer. So one thing is, for example, if you are, so first off, DeepMind did release their own Google Colab notebook. The disadvantage of the notebook is that it doesn't do any kind of a complex assembly. It only works with monomers. So it doesn't have any of this experimental stuff that I described to you guys today. It doesn't have the ability to sort of change the recycle. It only returns a single model, and it runs against only with MMC, with only a jackhammer as the alignment search method. And so this method is actually really slow. I mean, it's not super slow. It takes maybe 20, 30 minutes to make the alignment. So you could probably hit run and get coffee and come back. But if you're impatient, or in some cases, if you want more sequences, turns out MMSeq can sometimes find more sequences. So this is just the method from AlphaFold. So actually here, let me just open this up really quick so you can see what's going on here. So if you're like, you know what, I don't want to play with any knobs. I don't want to experiment with anything. I don't believe in MMSeqs. I just want to see what DeepMind tells me is the best possible structure at this point. Then this is the notebook for you. All you have to do is literally paste in the sequence here and literally just go through these cells and hit run, 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 and it will run through all those cells. But actually, one thing I should probably mention, just because there might be people here that are not familiar with the heck Google Colab is. So maybe I can give a quick introduction about this first. Google Colab is effectively Jupyter Notebook that's being hosted inside Google Drive. So the idea is like how you have Google Docs, how you have Google Slides, how you have Google everything. You can now think of it like now you have like Google Python or Google Jupyter Notebook. And so if you suddenly want to all of a sudden run Python code, you could literally just open up, like you can go to colabresearch.google and this will open up. And then if you press the connect button, you will actually get connected to one of Google's computers. So Google has Google Cloud. And it turns out if you just hit connect, it will randomly give you access to one of the free computers that they have sitting around that no one's using. By default, you get connected to one of the computers that only have a CPU. But it turns out what you could do is you click on runtime, change runtime type, and you can say, you know what, I actually want a computer that has a GPU on it or a computer that has a TPU on it. And the cool thing is Google will literally give you a free GPU for 12 hours and you can do with it whatever you want. And so here now I'm connected to the GPU session and I can literally just start typing code, say print hello world. I guess that's the typical thing people say. And suddenly you're running code on Notebook. And everything is directly connected to Google Drive and everything is saved. You can share the notebook with people, you can run. And so we use this platform. We're like, you know what, actually, since people can just get a free computer anytime they want, why don't we just build a notebook where we sort of filled in all these cells and have people just go through and click play, 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 and they don't actually have to type in these code and they'll actually have the whole thing executed for them. And now let's say, instead of running your own code, you now instead go to this GitHub repository that I just showed you, and you click on AlphaFold, you suddenly get the whole notebook and everything's already filled in with you. In fact, if you click on any of these little buttons here that says show code, you can see all the code that DeepMind has written for you and what each of these code blocks are doing. And of course, if you click on all these code, it becomes really, really scary because, but luckily they hid everything from you, so you don't have to see this. If we hit refresh, you'll get sort of a clean slate and everything is hidden away. Okay, but since we're here because of MMSeqs, we're not going to go through the DeepMind notebook. We're just going to go directly to the AlphaFold MMSeqs notebook. Okay, so whenever you open up the notebook, you get something like this. And what you have to do is press connect. And so this will connect to the notebook. And the thing that we do is we give you some options here. So first off, you can change the sequence that you want to model. You can give it a job name, and this could be useful because what happens is when the job is done, it will save your run into a directory with this job name. And so then it would be nice to organize those files, but you could just keep test the same if you want to. Then you can say which database you want to search against. In this case, we have mmseeks that search against UniRef plus the environmental database, but we also provide the option of saying, well, let's just search against UniRef. So that could be useful if you say, hey, I only want like things that have been well annotated and curated. But one warning here is that you might not actually find enough sequences. And finally, if you say, well, you know what, this is a de novo design protein, I don't expect to actually find any sequences, you can click single MSA, single sequence. And this could be kind of fun if you're like, you know what, I just want to know what physics AlphaFold learned. I don't care. I just want to disable any kind of MSA information. This could be useful. Or let's say you constructed your own custom MSA, you can hit custom. So for now, we'll just keep this constant. AlphaFold actually trained 5 separate AlphaFold models. And so by default, we will run all 5. But if you're impatient, you're like, I want to see the result, just run one model, you can hit 1. And the other thing is AlphaFold, well, the other thing they do is they take the final structure and then they send it off to sort of like a MD-like minimization of the structure to sort of fix a little bit of bond geometry. Turns out this almost doesn't move the structure at all, but just makes a few minutes more runtime. So if you click on that, it will do that. And then also, if you do want to use templates, so let's say if you want to steal coordinates from known structures in the PDB, you can click on this and it will do that. And finally, you can enable some experimental options where you say, well, I think this is a homo-oligomer structure, like there's multiple copies of this protein. You can set this to, in this case, I added up to option of 8. Okay, so once you set your parameters and you're happy with all those parameters, then you could go through and click on these cells one by one. But one thing that we try to make life easier for people is say, once you put all your features in here, you can just click runtime, hit run all. And then you agree that you're okay with this notebook running on your computer. On Google's computer. And what's happening now is that it got connected. And now it is installing all the dependencies. And this step takes maybe about 2 minutes. If you, within the same session, then change the sequence and then hit run again, it will actually skip these steps right away because they've already been done. And so the second sequence will run much faster, for example. So this right here is running, installing the dependencies. And if you're like, you know what, I want to know what's going on, you could always click on show code and see what's going on. But this is more like for advanced users, so you can just double click and hide that for now. Okay, so one question here is Chris asking, does use Amber use GPUs? Yes, it does. So it uses this library called OpenMM. And so it actually does install a special CUDA drivers that will allow it to use the GPUs. And the reason why I sort of have it turned off by default is because if you do press use Amber, it will actually download like a Conda environment in the back end, set up a bunch of libraries, and that actually takes a few more minutes to set up. Like here, you notice how it finished in like a few minutes. But had I use Amber, that would have taken another 5 minutes to install everything. Okay, so the first thing happened here is, so it finished installing dependencies. It imported all the libraries. It called mmseeks. And in this case, the sequence has previously been submitted. And so you got the answer almost instantaneously. But had you changed the sequence to something that's not been submitted before, this step would have taken up to four minutes to run. And so here it submitted the sequence to the server and it got back 1,935 sequences after filtering to remove any redundancy. So then these multiple sequence alignments get passed to the AlphaFold model. And now it's running each model one by one to predict the structure. So in this case, there's 5 models that were trained. And so we told it to make 5 predictions. So it's going to go through and make those 5 models. Okay, so while that's running, let me see if there's more questions. So one question is, for the MSA, how should the proteins be merged? We do provide a notebook that tries to do the merging for you. So right now we're going through this notebook here that was designed to predict monomers and homoligomers, and we'll search against the mmseeks database, has the ability to run with templates if you say yes. But we also have another notebook here that tries to predict protein-protein interactions. So given 2 proteins, in fact, let me just load this up while the other notebook is running. So I won't run this notebook just because I don't want to start another session, but it does have the ability to input 2 sequences. And actually, if you press this pair MSA option, what it will do is it will look at the 2 sequences and try to determine, are they coming from the same genome? And if they are coming from the same genome, it will try to concatenate them together. Okay, so now we're now at model 3. Yep. I mean,
we actually fiddled with the databases today. I was a bit worried that something might go wrong now, actually, in the demo.
Well, yes. Let's see. I guess while we're waiting for this, oh, actually, it looks like it's almost done. Let's see. Was there any other questions here? Supply. Oh, the question is it possible to supply your own templates? Technically, you can, but we don't provide that with the notebook right now. I mean, if that's an option that people want, I mean, it would be relatively trivial to add that option. Because one thing we could do is, the simplest thing is if you provide a PDB code, we could just retrieve that and just align it. Okay, so the run has finished, so I'll explain what these plots are now. So first off, this is the plot that I've shown you guys during the slides, where this is the number of positions, this is the predicted LDDT. And in this case, we ran 5 different models. And so it ran all 5 models, and it ranked all 5 models from highest confidence to lowest confidence. And you can sort of see all these 5 models. In this case, they're all pretty confident, all the 5 models. What you see on the left side is the sequence coverage. So at each position, how many sequences there are. And I think in the talk, we said we need at least 30. This one has way more than 30 sequences. So there's a lot of information here. Like if this number was, like, let's say if this was below 30, then there would be some concern because then the model might not have enough data to actually predict the structure. Then for every single model, there's 5 separate models that were predicted. We also have the predicted alignment error. And so this is a scale in angstroms. And so we want this to be as low as possible. And so you could think of this as sort of a metric that tells you for any given residues, the distance at which it predicted is really confident that that's the correct distance. If it's really high, in this case, it goes up to 30. Or so if you see anything red that tells you, okay, maybe I don't believe in that that residue interaction. Finally, once this is done, you will see the 3D model posted below. And in fact, the model is already colored by confidence. So blue being very, very confident. This is confident, okay, yellow, very low. In this case, it's very, very confident about this model, so it's all colored. And the other thing we could do is we can color it by chain so that we can see where the N and C terminus is. So if you change this option, hit play, it will return the same structure again, now colored by just blue to red N terminus to C terminus. Or you could say, hey, I'm just going to color it by chain. So it will all give it just the same color. If this was a protein complex, there would have been multiple copies and they would each have a different color. You can click here to see the side chains of the model. You can also click to see the main chains of the model. And so all looks good. And so this is the, once again, this is the same information as here and here, except now what you can do is you can say, hey, I want to see model number 2. And this will give you the information just for model 2 and this plot just for model 2. So it just makes it a little easier to see what's going on. And finally, once this is done, this it will zip all the information. This includes the A3M file that it made, all the 5 models. Had you pressed the use amber, it would have also given you like 5 models that were unrelaxed and the 5 models that were relaxed. So you can see both of them. And including the PNG images of showing the statistics. All these images are being saved, so you can load them up and look at them on your computer. And if anything was not clear, we do have a bunch of Q&A here telling you like what these metrics mean and so on. Okay, so and once everything gets saved, it will automatically just download the zip file to your computer. Sometimes people notice, like if you have an ad blocker, it might not download automatically. So if that ever happens to you, Google Colab lets you actually see the file system. So let's just zoom out a little bit. If you click on this little folder icon right here, you can actually see all the files that were created. And so the results are saved here. Like this is the job name result.zip. And so you just click on these little 3 buttons and hit download, and it'll actually download that file again. Or you're like, you know what, I don't want to download everything. You could just individually click on any of these guys. And in fact, if you click on any image, it will actually show up as an image on the side here. So you can see them on the side too. So this is how you would troubleshoot. Now, like now that we've finished this notebook, let's say if we go back up here and say, you know what, I have a reason to believe that this actually forms some kind of a oligomer, like there's multiple copies of this protein. You can scroll back up and say, well, I know this protein is, for example, a 6-mer. And so you can, once you hit that option, the easiest thing to do is to click runtime and run all. And you'll see right away, see how fast all these options ran, because it's already saved in the session. And then the only thing that's left to do is now to run the next model. And once this finishes running, you'll see the complex is now, and these will be all colored by the chain. Okay, I think there are some questions here. If I just want to get a good alignment and feeling lazy, can I get it from this notebook? I suppose you could, yes. So I think if you just want the alignment and you don't care about running AlphaFold, you could technically just run this cell, this cell, this cell, and this cell, and just stop here. And then if you click on this little folder, you'll see the alignment saved here. So this is .a3m. So this is the alignment. And you could just hit here and download that alignment to your computer. So that's if you just want the alignment for a reason and you don't care about AlphaFold. Yeah, so Julian is asking, can we expect the confidence of the monomer versus homoligomer to correlate with real life? So far, in all cases that we've tested, and I mean, this is just like handpicking a few cases, not like an extensive benchmark, it seems to be pretty well correlated between, like, if the molar actually forms a homoligomer, you actually see the 2 copies, and it's really confident about that interface. Whether or not we could actually predict how many copies there are, I think it really depends on the symmetry. What happens is if you have a cyclic symmetry, the larger the number, the more copies there are, you have these lever effect arm problems where, like, this interface is the same as this interface is the same as, like, these interfaces are really, really close to each other. So Gilliam asks, is there a way to bypass the 1,000 number of amino acids limit? So unfortunately, if you have a 16 gig GPU, there's not much you can do. I mean, at 16 gigs, it seems like the max you can do is maybe about 1,400 amino acids. If you do pay, there is an option to pay for Google Colab, and it'll give you higher priority. But once again, that doesn't like guarantee you any better resources. It just gives you higher priority. And often you only get a 16 gig GPU anyways, even if you do pay money. Like in this case, I think I have the Pro version, and all that does is just give me higher priority. One thing you could do is these notebooks, they're just normal Jupyter notebooks. So you could actually, if you do have your own cloud instance somewhere, you could potentially just load those notebooks in there and run it there with a larger GPU. So the question was DeepMind concerned we were encouraging the use of AlphaFold 2? I'm not sure how concerned they were. I think they were kind of, they were a little worried at the beginning, from what I could tell. I think initially when I first posted a Google Colab notebook, I just said, hey, look, I can predict de novo proteins where you only need a single sequence input. And I think they were a little worried that if somebody used this notebook and not all the features were enabled, and then they would say, oh, AlphaFold is not that good. And so I think that's probably part of the motivation why they decided to release their own notebook, because they're like, we want to make sure that everybody runs it the right way. But that being said, we've now validated the MMSeqs approach, and it actually produces alignments that give results that are equivalent to the DeepMind's approach, at least their Colab version. it's actually, their Colab is actually kind of interesting. The way they actually do it is they don't send the sequence off to a server. What they do is they create a virtual drive in RAM. They stream this big giant database into RAM, search it and delete it, and search it and deletes it. And so they're pretty much just streaming gigs and gigs of data into the Colab session and running jackhammer on each chunk one at a time. And that, of course, can take literally an hour for a large protein and maybe 20 minutes for a smaller protein. And it's one way to deal with the problem, but it would have taken much longer. But the way they got around that problem is by reducing the redundancy of the database. And so that's why it's still not as good as their full protocol that takes many, many hours to run. The way we solved the problem was to send the sequence off to the mmSeq server, run this super fancy algorithm that Martin just told you guys about, and get the sequences literally within minutes, and that sped up the whole procedure. So Alina is asking, do you think AlphaFold can deal with larger assemblies like protein cages? I think it should be able to. So I think the example that I showed, for example, in this, so it's able to form like this cage-looking structure for this homoligomeric assembly. So I suspect it should be able to also predict cages. But I mean, only one way to find out is to try to throw them all in there. I mean, the only limitation is the memory. So what happens is as soon as you put enough copies that you're now larger than 1,400 in length, you'll just run out of memory. But assuming maybe you can form part of the cage and see what happens. How do you train on protein structure templates with several conformational states? Also, can AlphaFold 2 and RosettaFold predict multiple conformational states? That's a good question. So, so all the different templates, I mean, all the conformations are being used as part of the training. And that might explain why sometimes you do get different conformations. So Gustavo pointed out that, so what happened was AlphaFold was trained with five effectively different seeds and slightly different number of sequences and so on. And it turns out that each of the 5 models returned by the model actually has a different conformation. So there's a possibility that each model sort of specialized maybe on different space. I mean, this could also just be somewhat stochastic, so I'm not sure how real this is at the moment. Okay, so let me check to see. Okay, so this almost finished running.
So just to follow up a little bit on the oligomer versus monomer. So you're saying as you go from like a dimer to tetramer to, as you go higher, the confidence increases, even though for some reason, even though it shouldn't, or I didn't get that.
Well, so I mean, it turns out it has to do with what kind of oligomer it is, homoligomer it is. So what I mean by that is, so this is an example where the interface between the 2 proteins, this is a homodimer, the interface doesn't have, I mean, there's just a symmetry going this way, but it doesn't continue making sort of like a big giant circle like some C symmetry. And so here you give it more than 2 copies, it effectively just makes 2 separate proteins, I mean 2 separate complexes. Because that interface, because it's anti-parallel and you're just pretty much locked into place and you cannot go any further for these 2 proteins. But for this protein, where what happens if I switch back between the 2, what happens is that because it has this cyclic symmetry, what happens is as you add another copy, it's able just to insert it in there. And I suspect the reason why this is true, if we go back to this Ryan's video, which I thought was really cool, if we look at this image here, so what it looks like is if you input the same protein multiple times into AlphaFold, what it does is it actually all the coordinates effectively start in the same location, and then it sort of unravels them into a circle. So imagine if you put like 10 copies here, it will continue going around and form maybe all 10 copies in a circle. If you put like 3 copies in here, it will go until it forms 3 copies. So I suspect this is why it does that, is because if it appears, I mean, this is kind of cool. It kind of appears that it learned some kind of symmetry, like it knows that there's some kind of symmetry going on here, and then it sort of rotates around that symmetry. And so if you have more copies than there should be, and might actually keep rotating, and then it will actually put, let's say, the 4th copy here if you had 4 copies, and maybe put the 5th copy there. So that's
But do you expect the confidence score to go up as you add more, sort of?
So this is, I mean, so I haven't done like a quantitative analysis on this yet, but like so for example, if you look at this one, just the correct answer, it does appear to be more blue on average than this one. So you can see it's becoming a little less confident. So it does appear that it got less confident, but I don't know if this is just happened to be true for this case. I mean, in reality, we'd want to do some kind of benchmarking. So one thing I should warn people is AlphaFold was not trained to predict complexes. This is just sort of like a post-discovery where people are like, hey, look, actually can't predict complexes. So we're using it for an unintended behavior. So this is completely experimental options. And how well it actually works is a little, is yet to be sort of benchmarked. There was some benchmarking. That being said, I believe I linked 2 papers here. And one, so there's 2 preprints here, one here and here, where they actually did benchmark protein peptide complex prediction on, and so I would recommend looking at these 2 papers if you want to see sort of more of a, so this is not like homoligomer protein, this is protein peptide, but it might give you some sense of how good it understand these things. Okay, so it looks like the thing that I just told you about finished. Okay, so now you'll see, so here what I did was I told AlphaFold that there's 6 copies of this protein. And it ran, and this time it ran a little longer because now you're effectively made the length 6 times larger. And now the confidence is over 6 copies, and so that's why you see these black lines here. And now you could see, if you actually zoom in on a little bit here, you could see that there's actually 6 copies in these little boxes, and you can see the confidence between each of those boxes. And if we go down, you can actually see now the homosexmer structure. So all the chains here, let me just hide the side chains and the main chains too. And let's just switch back to model 1, because that's the most confident one. And what's happening here is it'll tell you, okay, so these are the 5 models, and this is the average predicted LDDT. So I guess one thing I should stress is that it's important to look at the per position, I mean, at each position confidence, but also this is a very important, especially if you want to know like how confident it's about the domain, because it might be really confident about each domain, but it might not be confident about the interaction. And this is true for both large multi-domain proteins and complexes. As to, I think this metric will tell you how confident it is. So one question is, if we want to change the seed or if we want to change the number of recycles, do we need to change the source code? So you don't, if you just want to set a recycle number, you don't have to change the source code. It's just a configuration. You just have to change one configuration flag. But if you want to sort of implement this advanced thing that I talked about, and I think now that people are asking about, let me just show you. Okay, so this is sort of like our default notebook that uses MMSeqs, and it gives you sort of like the basic features that I talked about. But now let's say if you want to get a little fancy, you're like, you know what? I want to actually try changing the recycle number. I want to try different alignment methods. So this is where I would suggest the advanced method comes into play. So the advanced notebook is now maybe intended more for, I guess you could think of it, expert users. And so the idea is like same thing here. You can specify a sequence, specify the number of copies that you think are formed. But one thing that we added new here is that you can now technically pick MMSeqs, or you could say, you know what, I actually want to use DeepMind's jackhammer approach. So if you want to play around with both examples in the same notebook, you could try this one, or you could say, hey, I just want to have a single sequence. So that's one option that we added. And now this is where the whole recycling comes into play. So the idea is, actually, you know what? While I'm explaining this, let me just go ahead and I'll just set this up so it's running while we're talking. Okay, so the MMSeqs, this is the approach that we introduced today to you guys. But if you want to try jackhammer, you could. So there's some options here. Further, we also added this option for coverage filtering. By default, we turn it off, and I would not really recommend using it unless it finds way too many sequences. Let's say if it found like 100,000 sequences, what we found that requires a lot more GPU memory. So there's no reason to filter the sequences because the AlphaFold procedure is able to automatically filter the sequences. This is only useful if you found too many sequences to a point where the session just crashes. So it might help to say, well, you know what, let's just keep sequences that cover at least 25, 50, 75, or 90% of the total. Now for running AlphaFold, so it turns out, and here I tried to actually explain this in the text here, that AlphaFold actually trained 5 models, but then they took each of those 5 models and then further trained them, I guess you could say fine-tuned them to return a few extra metrics. And this is like the PAE plot. So the reason why we're able to make these predicted alignment error plots is because we're using these PTM models that output those for every single of the 5 models. But one thing we notice is if you don't have enough sequences, sometimes disabling this will give you slightly different variation of the models. So if you want sort of get like another variability of models, you can disable this. I mean, you won't get the PAE statistic anymore, but you might get slightly different models. And here I have now the option to specify the number of recycles. And if you set, for example, for 48, I mean, this is going to take a really long time. So if you do set a really large number, I would also recommend setting a threshold. So what the threshold is doing is it sort of runs each recycle. And as soon as it hits a point, so in this case, let's just set this to, let's say, 0.5. And just to make this a little more interesting, let's just set this to 4. So what will happen is I tell you, run up to 48 recycles. And so what it does, it will just keep looping recycle after recycle. And as soon as the difference between the previous recycle and the next recycle goes down to 0.5 angstroms overall distance, then it will stop. And so this is one way to potentially prevent this thing running forever or actually running up to 48. And so to make this extra stop thing, we did actually have to modify the AlphaFold source code to introduce that. If you don't want to modify the source code and you just want to set a threshold, it's actually just set up in the configurations. So this is going to look a little scary, but technically what you could do is when you first configure your model, you can specify the recycling here. And to specify this recycling does not require modifying any source code. You can just, whatever notebook or whatever script you're running, Colab with, running AlphaFold, you can just go in here and specify the number of recycles. This tolerance threshold is something that we added to the source code. So this is something you will actually have to apply a patch to the source code. So if you guys are curious, in the installation, what happens is we download the AlphaFold source code. Then I download a series of patches, and then I apply these patches to the models to be able to introduce this thresholding. So that's a modification we made to AlphaFold. Okay, so now what will happen is it will run. And the other thing we found is that changing the random seed sometimes makes a difference. Instead of letting you guys specify a seed, what we decided to do, well, if you say, well, let's say if you want to try like seed 1 through 10, you can just go in here, say, try seeds 1 through 10. And so it will actually run through all 10 seeds, and it will report to you every model and what seed number it tried. I mean, if you want to do more than 10, I just kind of got lazy and didn't want to type in all the numbers, but you can go in the source code and change this to 10 or 100 or something. Okay, so this is what this will do is we'll actually iterate through many different seeds, and it will save all the models, rank them, and give you the top 5. But it will let you download all the models. The other thing that we did is we added this option called Turbo, which tries to make AlphaFold run faster. And what this option does is, so it turns out that if you're using templates, like if you're using templates as input, it will actually, different models were trained with, some models were trained with templates, some models were trained without templates. And so you actually have to compile all 5 models. But if there's no templates, you can technically just compile one model and just swap in the weights. And so to make the notebook run faster, we just decided, we're going to compile one model and just swap in the weights. And so that makes it run extremely fast. And so, but there's also the possibility this hacking that we did to make things run faster might affect results. So far, we haven't seen it affect results at all. But if you're worried, you can always just turn this off and say, you know what, just give me the default behavior. I don't want Sergey's hacking in here. And so you can just turn this off and it'll run the same thing. And finally, if you actually want Amber to be run on all the models, you can click here and it'll run all the models. This is just an option. So what happens is the PAE score is saved as a big giant JSON file. And if you're planning to do a whole bunch of sampling, you don't want to run out of disk space, you can just say, you know what, I don't care about saving the statistics of each model. So you can just disable this. Okay, so now while this is running, you can see what's going on here. So the first model ran. It ran for 3 recycles, and it hit a tolerance of 0.22, and we set the tolerance to 0.5, so it stopped at 5 recycles. But the second model ran, and it actually needed 4 recycles before it hit the tolerance of 0.29 in this case. Here, it's for each model, it's returning to you the average predicted LDDT score. This is the same score we talked about, but it also gives you one additional output. This is the predicted TM score. So TM score is another metric that people often use to describe similarity between models. So if you're more used to the TM score metric, you can use that as well. So now it's running model 3, and eventually we'll run model 4, and so on. And what will happen is after it ran all the models, it will save all of them and then re-rank them and give you outputs. So this is what was added to the advanced notebook, is this ability to sort of maybe potentially introduce more variability. And one thing I should mention, that this is only really useful when you don't have enough data. Like if you have thousands of sequences, it turns out even running like literally with 1 recycle often just gives you the right answer. So what happens is when there's very low amount of data, what I'm beginning to suspect is this recycling is a way to effectively like sample. Like you're doing almost like abrilax, if you're familiar with that terminology from Rosetta. You're sort of just saying like running a trajectory and you're sampling. But if you have really good MSA and you got really good convolution information that it's able to extract, it's able to just get that structure like in almost instantaneously without having to sample. Yeah, so okay, it finished the 4th model now, and see, you can see here it ran for 3 cycle, 4, 4, and 3. So far what we see is that, yeah, so you can always just set it to a large number and set the threshold, and it will just run.
So the tolerance is half an angstrom. Does it make sense to set it that low?
I mean, it's probably not necessary, but I don't know. It's up to you. I mean, if you feel like half an angstrom is too much, you could set to 1. It does actually converge to, like, if you run it long enough, like in this case, it hit less than half an angstrom with like 4 recycles. I mean, I do think it's worth trying to get this as low as possible. I suspect if you don't let it run long enough, yeah, the solutions may not be as good. So whether or not it's a good default or not, I don't know. It's still up for. Okay, so it finished running all the models, and by default, so here's all the 5 models, and they're ranked from highest to lowest. And the way we name them is, okay, this is rank number 1. This is using model parameter 4, using the PTM option, which is specified here, with seed 0. So let's say if you do sample more than 1 sample, it will actually make many more variations and rank those guys. And finally, now we can observe these models to see what they look like. And this is the same thing that I showed you guys before. So this is, in this case, I think I told it that it's a homo oligomer with just 4 copies. And so here you can see it returned 4 copies. So one thing that's kind of interesting, and this is actually something that AlphaFold actually described in the paper, is even if you model a single copy, it seems to have some implicit understanding that it will form into a complex, and it looks like it almost prepositions itself ready to accept sort of like the next copy. And so there seems to be some implicit understanding that there is going to be more. Like it doesn't try, like in Rosetta, like if you try to model this, it will try to collapse this. Well, DeepMind will say, you know what? I know that this forms a higher order oligomer, so I'm gonna pre-position myself to sort of accept additional copies. Okay, so let me check the chat box just to see if there's any more questions. Oh, Philip B is asking, could you connect to your local Jupyter session? Yes, you definitely can. So if you click on this little arrow button here, you can actually connect to local runtime, and everything will run. So the idea is that you can actually launch a Jupyter session on your personal computer, and then you can take the local host port number and just type in the port number here, and it will actually connect to your local computer. I'm not sure if there's much reason to do that, because you could just literally just download this notebook and run it directly on Jupyter. But if you do feel like you want to have the experience of Google Colab, you could actually connect to, and actually the other thing I should mention is not just your local host, but you can even connect to your university's cluster. So you can actually launch a Jupyter session on your university cluster, get the port number and enter it into here, and it'll be able to connect to that session. So if you want to keep it all within the Colab experience. Okay, yep, so that's the advanced notebook. And I guess I think we're running out of time, so I will just say one last thing, I guess, because so far we've been only talking about AlphaFold, but we should give some love to RosettaFold. We do also have RosettaFold now as well. And if you click on RosettaFold, I mean, the notebook is almost identical. The only difference is it now actually runs RosettaFold. And so you run the sequence, and you can display the structure, and you'll get the structure and everything. One caveat, though, is that in order to run this on Google Colab, we do not include PyRosetta, and we have found that using PyRosetta to refine the structure further does actually help. This would be more like if you just want to sort of see roughly what the structure looks like, don't want to run the phone protocol. So we would actually advise people to use the Rebecca web server, which implements the full pipeline. You don't have to install anything. You don't have to use Google Colab. You don't have to worry about max residue links. You can just run the web server directly. This is more like if you want experimentation. You might wonder, like, why should you bother with RosettaFold if AlphaFold appears to be doing better? One thing that we do find is that sometimes when you don't have enough data, the different models, they sometimes specialize in different structure space. So sometimes what we found was like RosettaFold is able to predict some models really well that AlphaFold cannot when you have limited number of data, and vice versa. AlphaFold might be able to predict something that RosettaFold can't predict. So if you don't have enough data, because these models were sort of trained with like different seed, and in fact, RosettaFold is actually quite a lot of differences in the architecture, it might have actually specialized on something that AlphaFold did not specialize, and vice versa. So I think it's still worth trying to run your sequence, even if, but if you have like infinite amount of sequences, then I think AlphaFold will at this time will do better. But if you have limited number of data, I think it's worth running a RosettaFold just to see what it will give back to you, because it might actually give you a solution that lets you phase your data and so on. So it's like in the same spirit of how we're running 5 models with AlphaFold and changing the seed and so on. You can think of RosettaFold as sort of like this additional model that you can run and maybe get more structure variability.
Sorry, maybe you probably went over this and I missed it, but at some point, does MFSeeks tell you how many sequences it actually found in the MSA?
Yes. Yep. Yes. So after it ran, it will tell you how many sequences it found.
Oh, okay.
But also we provide this little graphic here that sort of tells you how many sequences. So in this case, total sequences. Right. So this will tell you for each position how many sequences it found. And also it'll tell you, as far as the sequences that it found, how close they are to the query. So if this was all purple, that means all the sequences are identical to the query. You can see here there's quite a large distribution of sequences, like some of them are far from the query, some of them are closer. So this graphic tells you that, but also we provide it as a number here.
And the final download also contains the MSA, so you always have your input MSA. So you should be able to reproduce your results with everything we provide in the zip file at the end for the download.
And I guess one last thing for those of you who are our surviving crowd, if you like that animation that I showed, I do provide a notebook that can recreate that animation for any given sequence. I mean, this notebook is sort of something I made today, so it's not very, let's say, human friendly, but it gives you all the code to make this animation at the end. This is something I killed like 3 hours of my today because I was trying to figure out how do you draw a structure with matplotlib. And also because it's a 3D structure, you have to draw these lines so that they're ordered by the Z dimension. So that way you draw the lines that are closer to you first and then draw the lines that are further away from you. So that way they don't appear to be overlapping. So this was a little bit overkill, but I was kind of proud once the final plot came through. And this is going to be only very useful if you're dealing with cases like de novo design proteins where there's no sequences and you want to run as many recycles as possible and you're kind of curious what those recycles are doing. I've written this function here, which is a little bit scary probably, but it goes through, aligns all the structures back to the final recycle unit, colors things, orders things by the Z dimension, and then it colors them by rainbow and plots all this stuff, and it makes the animation. So I think if I kept going at this, because at some point I started implementing some functions here to for the coloring. I was like, you know what? Actually, maybe I should color the lines behind darker so you have some shade. And so I added some shade functions here, but I ran a little bit out of time. Anyways.
Thanks so much for the really informative tutorial tonight, Sergey. Thanks, Sergey.
Of course, no problem.
Thank you as well for coming out and for teaching us about MSAs and for making this process fast enough to actually run in Colab. And thanks everyone for coming out tonight.
Yep.
Yeah.